Bismillah ar-Rahman ar-Rahim. Bismillah ar-Rahman ar-Rahim. Good day and Assalamu alaikum, everybody. Ladies and gentlemen, I'm Hazima Mozam and I welcome you to another exciting lecture under Comstech Distinguished Scholar Speaker Series. We are very pleased and delighted to have Professor Dr. Keykavous with us. Professor Dr. Keykavous is an Associate Dean of Research, Graduate Studies, and Global Affairs. He is Professor of Medicinal Chemistry and Pharmacology at Chapman University, United States, America. Today, the title of his talk is Cyclic Peptides, Anti-Cancer Drug Delivery. And with this, I would like to thank our speaker, as well as request Professor Dr. Iqbal Chaudhary, Coordinator General Comstech and Director ICCPS, to introduce and welcome our guest speaker for today. Over to you, sir.
Bismillah ar-Rahim. Assalamu alaikum. Good morning and good afternoon, ladies and gentlemen, boys and girls, lots of regions and different age group of participants among a very crowded seminar room. And we're extremely pleased and fortunate to have a very distinguished speaker today. Comstech, being an organization responsible of science and technology cooperation within the 57 OIC member state, is responsible of many activities, and we have taken several initiatives. One of the initiatives in the context of reduced mobility is to reach out to very distinguished scholars all across the world and request them to engage themselves into this extremely exciting exercise of presenting the best of what science and technology offers. So this particular series of lecture is dedicated to only the best in the world in various fields. And we are very fortunate to have a very distinguished scholar today, who is also happens to be a very dear friend and a long-time research collaborator. We have had the honor of meeting each other at different places. Professor Prang is an Associate Dean of Research, Innovation and Global Affairs and a full Professor of Medicinal Chemistry and Pharmacology at the Chapman School of Pharmacy in Irvine, California. He earned his Doctor of Pharmacy from Tehran University of Iran. Tehran University, as you know, is a very prestigious institution. And from then, he received his PhD in medicinal chemistry from the Faculty of Pharmacy at the University of Alberta in 1997, followed by a postdoctoral study in the field of solid phase organic synthesis in the Department of Chemistry. He pursued additional postdoctoral studies at Rockefeller University, New York, and then moved on to Johns Hopkins University in Baltimore, focusing on bioorganic chemistry. He joined the University of Rhode Island in October 2000 and soon became full professor. He served as the program coordinator of Rhode Island IDEA Network of Biomedical Research, a very prestigious initiative which was sponsored by National Institute of Health. And he was responsible of leading this major program between 2012 and 2013. He then joined Chapman University to establish the School of Pharmacy. Dr. Prang's research can be described as applying synthetic organic chemistry, skills of synthetic organic chemistry to solving problems in biology. His specific areas currently under investigation include using peptides as cell-penetrating molecular transporters in drug delivery, designing protein kinase inhibitors, developing multifunctional antiviral, anti-cancer, and antibacterial agents, and designing peptide nanomaterial for nanomedicine. So you can see that he is actually covering a whole range of pharmaceutical sciences and medicinal chemistry. One major area currently under investigation is to design peptide nanomaterial for application and drug delivery, which is certainly cutting edge. The objective of this project is to design and evaluate peptide nanomaterial as cell-penetrating nuclear targeting agents or molecular transporters or bioactive cell impermeable compound. Study will document the potential for new hybrid peptide drug samples that may be used for non-covalent or covalent targeted delivery of bioactive compounds. So certainly an area of extreme importance. He has authored over 210 research papers, 13 pending patents. He has already successfully obtained 10 patents and more than 180 meeting abstracts. So he's certainly one of those scientists who has not only worked at the level of establishing good quality education institution, contributed in high quality teaching, but also pursued a very high quality research. With this multifaceted contribution, Professor Parang is certainly among the world leaders in the field of medicinal chemistry, and it is indeed an honor for me to introduce an expert and a dear friend today in this very important workshop series. Thank you very much.
And thank you so much, Dr. Chaudhary. It's great to see you. Thank you for extensive introduction. And I also appreciate your invitation. I'm very delighted to present today. With that, let me share my screen with you so we can get started. Can you see my screen?
Yes.
Okay. So today I'm going to talk about cyclic peptide and the application as anti-cancer drug delivery agent. So for those of you from different area of the world, I'm located at Chapman University. It's in the Southern California. And whenever you're visiting Orange County and Southern California, please let me know. I'd be delighted to be your host. So let me go to some information that many of you are aware of. So as you know, cancer is the leading cause of death. If you imagine for COVID-19, we had about, so far we have about 188 million people infected and only 4 million people dead. So we're talking about 2% fatal pandemic. In terms of cancer, we have 19.3 million people had cancer in 2020 and 9.9 million dead. So we're talking about around 51% dead. So, and as you know, this is a very complicated disease and is involved in different organs. This year, breast cancer surpassed the lung cancer as a number of the cases, around 2.3 million, a little bit more than lung cancer, 2.1 million. And still we have mortality rate for lung cancer is much higher than all other cancer. And this is continue to be a major problem in the world. If we don't call it a silent pandemic, you know, a lot of attention to COVID-19, but cancer is continue to be a major issue for many people. And as we all know, we have family, friends, and relatives who have lost their life in cancer. In terms of treatment, as you are all aware, we have a major treatment is chemotherapy, but there are many other ways to treat cancer, including surgery, radiation, and targeted therapy is a new area that has been developed, and also immunotherapy. And many of these approaches are used in combination. So we don't have one optimal way to treat cancer, and sometimes we have to use even combination of multiple drugs in chemotherapy. We don't usually use one single drug, or we have to use different strategies like radiation therapy, chemotherapy, and surgery at the same time. So still, chemotherapy continue to be the major strategy to destroy cancer cells. It can be used as the only treatment option at the beginning, but also it can be used to shrink the tumor before radiation, and or it can be used after radiation and surgery to get rid of all the remaining tumor tissue and tumor cells. It can be used for recurrent cancer and metastatic cancer. So chemotherapy is still one of the major strategies that we use for cancer. And I'm sure all of you know the major problem with chemotherapeutic drugs are the selectivity and occurrence of the side effect in the patient, because many of these drugs are cytotoxic drugs and also affect the normal cell in the body. So in terms of drug delivery agent, there are many different drug delivery agents have been developed, and we're talking about liposome, that these are a spherical phospholipid bilayer, and you can have hydrophobic and hydrophilic drug loaded in liposome. The major problem with the liposome is that they have limited stability and they cause leakage. So there are also a lot of polymer has been developed, like polylactic acid, glycolic acid. PLGA is one of them that can be used for application for drug delivery. Another problem with this kind of compound is they are degraded and generate acid in the body and cause acidic environment. And sometimes acidic environment is desirable for cancer cells. And also solid lipid nanoparticles are more preferred compared to liposome and polylactic acid. So you can have a lipid nanoparticle, you can load the anti-cancer drug inside the lipid nanoparticle. The major problem with solid nanoparticle is they have a very low loading of the compound. So another application, that strategy that has been recently considered is cell-penetrating peptide. Cell-penetrating peptide are kind of peptide that they can pass the cell membrane. And peptide, as you know, there are compounds that are composed of different type of amino acid. And we have 20 different natural amino acid, and we have many, many more unnatural amino acid. So if you want to divide cell-penetrating peptide, there are different categories. We can have cationic, hydrophobic, and amphipathic based on the physical chemical property. Also, in terms of the structure, we can have linear cell-penetrating peptide or cyclic cell-penetrating peptide. So I'm going to talk about the differential advantage of these two. Also, they can be derived from the protein, they can be synthetic, or they can be a mixture of synthetic and natural at the same time. But in general, they can have multiple applications for drug delivery. If you look at extracellular parts of the cells and intracellular cancer cells, you know, we can have cell-penetrating peptide or CPP alone and be used for drug delivery. They can be part of the nanoparticle for drug delivery. They can be used for delivery of nucleic acid like siRNA, or they can be part of the liposome and polymers at the same time for drug delivery. They can be conjugated with radioactive nuclide for imaging, and they can be used as a cargo for delivery of other peptides. So there is multiple applications for cell-penetrating peptide that have been reported in the literature. But in terms of the linear cell-penetrating peptide, these are very short number of amino acid. We're talking about 6 to 20 amino acid, and they have very good high internalization, and usually they have amphiphilic property, means they have hydrophobic and positive charge at the same time. And they can be biodegradable and easily eliminated from the body, and most of the time they are composed of natural amino acid. The synthesis is very easy, and you can use solid-based peptide synthesis and also functionalize them. The major problem with the linear cell-penetrating peptide is toxicity, very short half-life, and they are not very selective, okay? So that's caused the problem that if you want to use them for animal study, they can be eliminated quickly. Some of the reported cell-penetrating peptides are TAT, PEP1, polyarginine, and many other peptides that these are usually have polyarginine residue or hydrophobic tryptophan residue or a mixture of both. So you can have arginine or tryptophan. So tryptophan is represented by W and arginine represented by R. So that's why even linear cell-penetrating peptide has been around for a long time. They haven't been very successful for drug delivery, and most of the time they are used for transfecting agent in cell-based assay because of the toxicity and also short half-life. So what we are interested is mostly cyclic cell-penetrating peptide. So these are cyclic peptides that compose of hydrophobic and positively charged residue. The reason they are more attractive because they have a higher stability toward the proteases, so they can survive the harsh condition of the protease and peptidases that we have in our body. And also they have a higher cell permeability compared to the linear counterpart, and they exhibit endosomal uptake. So many of the peptides that we have in our body, first of all, they will be degraded quickly. But those cell-penetrating peptides also, they may undergo endosomal uptake. Endosome is a way that the peptide will be encapsulated inside the endosomal package and will be trapped inside the cytoplasma, and they cannot escape. So we have to find a way to bypass the endosome and enhance the endosomal escape if we want to deliver any drug to the cell. So in terms of my research, with this introduction, I want to give you some general research design that I've been involved with. So first of all, we are interested in two amino acids. One is tryptophan, that we call it W, and arginine, that is R group. So arginine is a very positive, highly positively charged peptide, a basic amino acid. It has a guanidine side chain, and tryptophan has an indole side chain and is a very hydrophobic amino acid. So we were thinking to design peptide of combination of these that can be used as a delivery agent. So first of all, we developed some peptides that are linear, that alternate hydrophobic and hydrophilic residue like this structure. Or we have a hydrophilic residue like arginine attached to two tail of hydrophobic residue. Again, these are linear peptides, and we have been using them for the cell-based assay. And also many of these deliver the cargo to the cytoplasma, and they are very effective. But again, the application of this in the animal study and in vivo will be very challenging. So we became interested in combination of cyclic linear peptide or cyclic peptide or only cyclic peptide. So you can have a hydrophilic head and hydrophobic tail, or hydrophobic head and hydrophilic tail, or you can have a cyclic peptide that is fully hydrophilic, or one peptide, cyclic peptide that is fully hydrophobic. You can have a head with two legs, could be hydrophilic head and two hydrophobic tail. And you can have hydrophilic and hydrophobic alternate, or you can have all the hydrophobic in one side, all the hydrophilic in one other side. So as you can imagine, there is a huge number of possibilities that we can test, and actually we have tested many of these as a drug delivery tool. And I'm not able to cover all of this, so I'm going to highlight a few class of this compound. So interestingly, when we have a cyclic peptide like this that have hydrophilic and hydrophobic alternate residue, that like arginine and tryptophan alternated in the cyclic peptide, this kind of cyclic peptide can act as a drug delivery agent. But if you put all the hydrophilic in one side and all the hydrophobic in another side, you get an antibacterial agent. So the same number of amino acid, the same amino acid, but the sequence is different. In one, we have all the hydrophilic and hydrophobic in two opposite sides, and in one alternate. And one of them can act as a drug delivery agent, one is as an antibacterial agent. So I'm not going to talk about antibacterial agent. This is a huge area of research we've been doing, and we have synthesized hundreds of peptides as antibacterial agent. And we have very good lead compound against broad spectrum multi-drug resistant bacteria. And we have done some also animal study in infectious animals. So today I'm going to talk more about the drug delivery agent as an anti-cancer drug delivery of different compounds. So let me talk about just this simple peptide that have 4 arginine and 4 tryptophan, and we have used this in many different applications. The story goes back in 2011, we realized this peptide actually goes to the nucleus of the cancer cell, and it can deliver different cancer cell anti-cancer agent to the nucleus. And then we find out this peptide actually have different other applications. You can conjugate it with the different drugs like doxorubicin. It can have protein tyrosine kinase inhibitory activity. And we already have some idea what's the mechanism of this. We also conjugate this peptide with different anti-cancer drugs, also natural products. And we have used this peptide for nucleic acid delivery like siRNA. And recently we have used similar compound for delivery of CRISPR. And also many of negatively charged molecules like phosphopeptide that cannot be delivered into the cell. And we realized actually this peptide actually can generate gold nanoparticles and cap with the gold and be able to deliver different type of drugs. And also for the delivery of proteins, we have used some of this derivative of this peptide, and they can be used as a stabilizing agent for protein. So there is multiple applications you can have only with one peptide. So what is the process that this peptide can be like a kind of magic bullet for different applications? So we realized that this peptide that contains alternate tryptophan and arginine residue, it generates a nanoparticle. And as you can see, a small particle are self-assembled with each other, generate larger particle, and these large particle even self-assemble and generate even larger particle. So this process continues. The way it works that you have a hydrophilic and hydrophobic residue, in two opposite sides. And these hydrophobic residue love each other and they want to self-assemble with each other. And hydrophilic residue, that is arginine residue, they want to be exposed to the water molecule. And if you have a drug you want to encapsulate, especially if you have a hydrophobic drug, you can encapsulate it, and this nanoparticle, self-assembled nanoparticle, can deliver the hydrophobic drug inside the cell. So it's similar to what you have for Trojan horse, that you can have a hidden hydrophobic molecule inside the host and pass the phospholipid bilayer and deliver it inside the cell. So let me tell you a little bit data about this. So this is one anti-HIV drug, lamivudine. So we wanted to see how we discovered this peptide. So we use a cargo molecule that is anti-HIV and attach it to a fluorescence molecule. So we can track it inside the cell. And we designed many different cyclic peptides, not only WR4, but we use different hydrophobic amino acid, hydrophilic amino acid like lysine and phenylalanine, or even negatively charged cyclic peptide, and so on. So we tested all these compounds with these cargo drugs, and we physically mixed them. And among all of these, only WR4 and WR5 were able to improve the uptake of this compound. So we eliminated all these compounds and we became interested in tryptophan and arginine cyclic peptide. So this was delivered in the blood cell, leukemia cell, and we have like maybe 5 or 6-fold more uptake. And if we do the fluorescence study, if we use only the parent drug that is fluorescent-labeled, we don't see much uptake. But in the presence of WR4 and WR5, we see green fluorescence inside the cell. So these peptides were able to deliver fluorescent-labeled drug intracellularly. And if we look at confocal microscope of another cargo molecule, so we use a fluorescence-labeled phosphopeptide. Phosphopeptides are very cell impermeable. They cannot pass the membrane. So if this is green fluorescence, this is stain we use for the dyeing the nucleus, DAPI. The Texas red is used for dyeing staining the membrane. And if you merge all of this, we don't see any green fluorescence inside the cell. But if in the presence of cyclic peptide, first of all, we see the cells have been incubated with this fluorescence-labeled phosphopeptide, they become green. And then if we merge all of this, we can see the green fluorescence inside the cell. And if we look at a little bit, this is a phosphopeptide that we talking about, the green fluorescence. And I think I have here, if you use only phosphopeptide, you don't see any fluorescence inside the cell. So I was going to show you a video, but let me see. Okay, yeah. So you can see if you look at the three-dimensional cell demonstration of confocal microscopy, you don't see any fluorescence inside the cell. So you can see the nucleus that is blue and the membrane that is red. Okay, so if we incubate the same cell in the presence of cyclic peptide and phosphopeptide, you can easily see the green fluorescence inside the cell. Again, you can look at the three-dimensional confocal microscope, and you can see that the green fluorescence is actually inside the cytoplasm in terms of the phosphopeptide. So this peptide was able to deliver phosphopeptide inside the cell, and phosphopeptide, as I mentioned, are completely cell impermeable. In terms of toxicity, you can see that these peptides are quite safe, up to 100 micromolar. So we have a very good cell viability, and we have tested different type of cell line, including normal cell, and we don't see much toxicity, and cell viability is more than 95% or above that. So another way we wanted to see where the cyclic peptide goes, we can track where the cargo goes. What about the cyclic peptide? So we labeled the cyclic peptide with the fluorescent. So we have a fluorescent, and we incubated different cancer cells with this, including ovarian cancer, colon cancer, and breast cancer. So this is the green shows that the labeled peptide. This is the nucleus, DAPI, and if we merge them, everywhere that is green is also blue. So it means that this peptide actually goes to the nucleus of the cell. So this is a major application because many of the cell-penetrating peptides, they trap inside the endosome and they cannot come out of the endosome, or they may be able to deliver a compound inside the cell, but then if they trap inside the endosome, they are not able to deliver them to nucleus or other parts of the cell. So this is a nuclear delivery agent. Actually, we can use it for anti-cancer drug delivery. So now what we can do with this cyclic peptide for anti-cancer drug delivery. So we selected doxorubicin, and you are familiar with doxorubicin. It's a red fluorescent compound, and it has anthracycline, and it has a glycoside attached to the final link. So we have tetracyclic compound. This compound has interesting mechanism. Is actually intercalate with DNA. This is one of the mechanisms. It also inhibits the topoisomerase 2. Topoisomerase is an enzyme that actually nick the DNA. So when the DNA are coiled, they cannot undergo transcription. So this enzyme has been designed to cut the DNA and uncoil it and then reseal it. So doxorubicin actually and block the resealing of the DNA. So we have a kind of cut DNA that cannot undergo transcription. Also, it has another mechanism that causes oxidative stress, and it generates that can cause problem in transcription. But then there are a problem with this doxorubicin. This is a very active anti-cancer drug, active against many cancers. The major problem is cardiotoxicity and also kidney toxicity. But also there are many doxorubicin resistant cancer cells that after application, the cancer cells develop resistance. So how we can avoid this problem, cardiotoxicity and doxorubicin resistant bypass? So our thinking was that maybe we can use a cyclic peptide to avoid that. So if we look at the doxorubicin structure, you have a DNA intercalating agent, you have a glycoside, and also improve the water solubility, and then you have a topoisomerase 2 interacting domain. So where we want to attach our peptide. So we thought that many of these functional groups are important for interacting with DNA. So we selected this OH group and attach it to a linker and then use a conjugation with our peptide. Again, for doxorubicin resistant, one of the major problems, because there is a lot of efflux protein, one of them is PGP protein. We have also multi-drug resistant associated protein and breast cancer resistant protein. So all of these, especially in several cancers like ovarian cancer, we have efflux of the doxorubicin from the cancer cell. At the beginning it worked, but after a while you see complete lack of activity. Also, as I mentioned, doxorubicin causes the cardiotoxicity. And the major reason as the generation of this quinone that can interact with the oxygen and generate superoxide oxygen, and then radical OH, that is the Fenton reaction. That is radical is very active and can cause a lot of problem in cardiac tissue. So this is a toxicity issue for heart, but then the mechanism, as I told you, it interacts with the topoisomerase and kills the cancer cell by inhibiting topoisomerase by different mechanisms. So we have two major problems, cardiotoxicity and resistance. So we developed this peptide conjugate with doxorubicin through a linker, cyclic peptide, and we look at the uptake of this. So if you have doxorubicin in ovarian cancer, it can get into cancer cell, and it has fluorescence, and you can see it. The conjugate also can get into the cancer cell. After 24 hours, we don't see much retention of doxorubicin alone inside the cancer cell, but the conjugate have much higher retention inside the cancer cell. And so that was where the story started. So we started to think about developing hybrid cyclic linear peptide. So we have a head of arginine group in cyclic peptide and tryptophan residue and attach it to a linker to doxorubicin. It can get to ovarian cancer cell, but interestingly, the compound cannot get into the heart cell. So it was very selective against cancer cell. And if we look at the heart cell activity, antiproliferative activity of doxorubicin is here, and it can kill the cancer cell. But then the conjugate, even at 10 micromolar, it doesn't have much toxicity in the cancer cell, in the heart cell. The same thing for kidney cell. The doxorubicin is very toxic against kidney cell, but the conjugate was not very toxic against kidney cell. And we show that in the microscopy data. So if we have a doxorubicin, it can easily go to heart cell. But when you have the conjugate, we don't see much inside the heart cell. On the other hand, the compounds are very active against cancer cells. So if you have leukemia cell, colon cancer, stomach cancer, so doxorubicin is quite active against cancer cell, but also our conjugate is quite active against cancer cell, even at 5 micromolar after 72 hours. The same thing in the stomach cancer cell. You see doxorubicin is toxic and our conjugate is also toxic. So it's very selective against cancer cell, but not heart cell. And if you look at the fluorescence microscopy of doxorubicin in ovarian cancer compared to the conjugate, you see very high concentration of the conjugate inside the cancer cell, ovarian cancer cell. So if we look at the resistance profile of this compound, if you have free doxorubicin tested against wild type breast cancer, it has a LC50 of 0.45 micromolar. Our conjugate, which shows comparable activity against wild type. If you look at the resistance cell, you see free doxorubicin doesn't have much activity against resistance cell, but the conjugate is significantly more potent than free doxorubicin against resistance cell. Another resistance cell is a sarcoma that has an efflux pump mechanism. Again, free doxorubicin is not very active and conjugate is active. And we can see this also in this curve. This blue line shows the activity of free doxorubicin and conjugate in wild type. And this solid line, green and red, shows the activity of doxorubicin in resistance cell. It's not very active. And this comparable counterpart with the conjugate against resistant cells that are much more active compared to doxorubicin. So you can see also this in confocal microscopy. So if you have a free doxorubicin, it doesn't get into the resistant cells, so you don't have much uptake. Conjugate not only get to the wild type, but it also get into the resistant cells and also get to resistant cells that have efflux pump mechanism. So this is a triple negative breast cancer cell, and this is a sarcoma cell that have efflux pump mechanism. So there are also different uptake mechanisms. So we can have direct penetration of cell penetrating peptide, or you can have endocytosis. And as I told you, endocytosis is a major uptake mechanism, but it has a problem. And we have different type of clathrin-mediated endocytosis, or micropinocytosis, or keykavous-mediated endocytosis. So there are different type of vesicles that cause endocytosis. There are inhibitors of endocytosis mechanism like chlorpromazine and chloroquine. I'm sure you all heard about hydroxychloroquine, that people thought it can be used for COVID-19 to generate endocytosis inhibitor for interaction with a spike protein in SARS-CoV-2, but it was not very effective. But nystatin and methyl beta cyclodextrin also inhibit Keykavous-mediated endocytosis. So when we use our conjugate and look at the uptake in the presence of the different endocytosis inhibitor, we don't see significant reduction. Chlorpromazine and methyl beta cyclodextrin decrease the cellular uptake. But again, this could be a mixed mechanism of uptake, so partial inhibition, but not complete. So we believe that the peptide conjugate get into the cell through the direct translocation, and in the presence of different inhibitor, we still see significant uptake inside the cell. So the next part, I don't know how much time I have, so I'm trying to be concerned about your time. So if you have a cyclic peptide like WR5 and increase the size, WR6, WR7, WR8, and WR9, make it larger. And when we increase the size of the cyclic peptide, we realize that it's improving the cellular uptake significantly, especially for WR9 for the delivery of phosphopeptide. So we became interested in WR9 and conjugated with doxorubicin, the same approach we use for WR5. And the same thing, we saw that this compound is very active against cancer cells, like breast cancer cell, even more potent than doxorubicin. And in different type of breast cancer cells that even at 1 micromolar, 5 micromolar was more potent than doxorubicin at the same concentration. And if we look at the uptake of doxorubicin and compare it with the doxorubicin conjugate, we have much higher uptake compared to doxorubicin in breast cancer cell. If we use the resistant cell again, doxorubicin doesn't show any anti-proliferative activity against DOX-resistant cells, but our compound was significantly more potent against resistant cells. And again, we can see that the DOX actually doesn't get into the resistant cell, but the conjugate was able to get into the resistant cell. And also, again, we tested this compound in presence of different endocytosis inhibitors, and we didn't see any inhibition of the endocytosis. So that's another advantage. So we have a drug delivery agent that bypasses the endocytosis. We look at the hydrolysis of this conjugate. So at different time periods, in the 4-hour, 12-hour, 24 hours, and 48 hours, it's slowly converted. This is cell-based assay. So DOX conjugate is converted to the free doxorubicin, and after 48 hours, almost all of the compound has been converted to free doxorubicin. The half-life of this was around 18, 19 hours. That is acceptable for in vivo study. So in general, if we want to have an optimal drug delivery agent, you want to have a targeting moiety, and we can have an imaging agent, and we can encapsulate the therapeutic agent. So we're talking about selectivity to cancer cells. So we need to have some targeting moiety that can target cancer cells. So we selected folic acid. Folic acid, there are a lot of folic acid receptor overexpressed in different cancer cells. And also we attached PEG, polyethylene glycol, to improve the circulation of the peptide and improve the stability. And then we added the imaging agents like fluorescent tag, like Alexa-Fluor, to be able to monitor the drugs, monitor the delivery agent. So this is a peptide we attach to a folate to have targeting. We have a PEG to improve the circulation time, and then we have a tracking agent like CY55. And we added this conjugate we injected to animals that have two different types of tumor. On the left-hand side, there are tumors that doesn't express the folic acid receptor. On the right-hand side, this has folic acid receptor expression, HeLa cell. And then we look at the images. Most of the compound actually localized inside the tumor that overexpress folic acid. And we can separate different organ and also different tumor. And you see we have high localization inside the tumor. Also, we wanted to have a drug. So last slide, I showed you a delivery agent with the folic acid and also imaging agent, but we wanted to also have doxorubicin. So we attached doxorubicin, conjugate, PEG, and folic acid, and also imaging agent. So we have imaging, targeting, and drug and delivery agent, and also we have PEG to improve the stability. So we did some animal study, different time period, and we injected to animals and look at the localization in the xenograft tumor model, and we were able to see within 1 hour, most of the compound was inside the tumor, and we didn't see much in other organs. There was some in the bladder also. And if we look at this picture, let me show you the video. So if you see this 1 hour, we see most of the compound inside the tumor, and then it released into the bladder. After 3 hours, most of the compound actually has been eliminated to the bladder and getting released, eliminated. So we get some selectivity in the tumor within 1 hour, and then we have increased elimination from the blood. So in conclusion, we have designed many different cyclic peptide platforms containing tryptophan and arginine, and these are self-assembled into the nanostructure, and they can be used as molecular transporter. And we have many, many different platforms that haven't been published yet. Hopefully, we will see some of this in next few months. And some of these have been used for delivery of hydrophobic drug, and some of them have also been used for delivery of negatively charged phosphopeptide and siRNA at the same time. So these are some of my postdoc and colleagues and some of the students who graduated. And many other people, Dr. Rakesh Tiwari, Dr. Dindian Manda, Sandeep Lohan, and Sagar Muzaffari was involved in the hybrid cyclic peptide, and Paul was involved in many drug conjugates, and Riley was involved in siRNA delivery. And also funding from different agencies, including American Cancer Society, NIH, and many other funding agencies. With that, I open the floor for the question. I hope I'm not over time, so I'll be happy
No.
to answer any question you may have.
Thank you very much, sir, for a very insightful and interesting lecture. So we have some question and answers, questions in the chat box. I'll read them to you for your convenience.
Yeah, I can hear you.
Okay. So the first question is from Ms. Prayal Ashok. She's asking, what if some small proteins intrinsically possesses more amount of arginine and tryptophan? So we can have these activities or improved drug abilities. So your comment on it.
Okay, so there are arginine and tryptophan in many proteins, but they are not cell permeable because the sequence of this peptide has to be in such a way that they can interact with the membrane. On the other hand, we have used some of these peptides for delivery of proteins, including green fluorescent protein. So if you're talking about protein or peptide, if you're talking about peptide, arginine and tryptophan have to be in a specific alternate or a specific format to be able to deliver different cargo. But protein alone that have arginine and tryptophan, they cannot get into the cell by themselves. So we need always a carrier to do that.
Right.
Did I understand your question correctly?
Yeah, I think that was what she was asking for. She has two more questions. And both of them are linked probably. One is, kindly tell the rough estimates of molecular weight of these peptides. I think she's referring to cyclic peptides. And kindly also make comment on how cyclic protein achieve their 3D conformation if they possess. Okay,
the molecular weight of this peptide, usually peptides, they have a molecular weight, amino acid molecular weight of 170 to 200. So if we're talking about 10 amino acid, we're talking about around 2,000 molecular weight. This, as you can imagine, these are not a drug-like molecule. There are, if you're thinking about Lipinski rule and low molecular weight less than 500, these are not. These are just for delivery agent. Again, physical chemical property of this compound, they are amphiphilic. They have positive charge and hydrophobic residue. They self-assemble with each other. And they have different application because we're talking about generating nanoparticle. And nanoparticle, as you know, is used in nanomedicine, and they don't follow the Lipinski rule. So we're talking about 1 to 100 nanometer size of this nanoparticle for drug delivery.
Right. So we have another question from Dr. Abbas Hassan. He's asking, I understand that the cyclic peptides are compatible with the biological systems, but their synthesis is somewhat very difficult. Can we use unnatural or beta amino acid based peptides for the same purpose? However, so that their synthesis will be much easier. Your comment on it.
Okay, so cyclic peptide, actually, you're right, they can be difficult to synthesize, but the way we synthesize this, so you make the linear peptide and then you attach the head of the snake to the tail of a snake. So, and if you think about head and tail, they have to find each other. But if you have a very concentrated solution of peptide, the head of one peptide may conjugate with the tail of another linear peptide. So the way to do the cyclization, you have to always use a dilute solution. So the molecule cannot find each other, but only can cyclize by themselves. In terms of unnatural amino acid and using other type of peptide bond, yes, it is possible. The concept is the same. So you just have to make the linear peptide and cyclize it. But eventually, the technical challenge that is you make sure you have a very dilute solution when you do cyclization, so you don't get dimer and trimer and of the linear peptide, but you only get the cyclic peptide. So the synthesis is based on solid phase synthesis. You make the linear peptide, you cleave it from the resin, and then you cyclize head to tail. But it doesn't matter what type of peptide bond you have, the concept will be the same.
Right. We have another question from Ms. Paryal Ashraf. She's asking, you mentioned that the cyclic peptides may overcome protease activity, but how they evade HCl effect of the stomach? I am asking in context, if some peptides have been successfully approved as drug, we have concern of their oral deliverance of drug.
So when we say they are more resistant to protease, it doesn't mean they don't degrade, but they have a longer half-life. But there are 50 cyclic peptide drugs on the market already. So, and you already know some of them, like cyclosporine, daptomycin, vancomycin. So there are many cyclic peptides that they are pure cyclic peptide, or they are combination with other residue like carbohydrate. So it's not like they don't degrade, they will be degraded, but they have a longer half-life, and the protease has a hard time to cleave the rigid structure. But eventually we have many, many different proteases in our body, and some of the amino acids in the cyclic peptide are more sensitive to protease compared to others. So it depends on the sequence of the amino acid. Here we don't have exopeptidase and endopeptidase because the peptide is cyclic. But then eventually we have many other proteases in our body that they will cleave this, and they will be metabolized and they will be eliminated from the body. Did I answer your question? Yeah.
Yeah, hopefully so. And we have, so we can take, we're running out of shortened time. So I'll take two short questions. One is from Mr. Nadeem Altaf. If a comparison is made between synthetic cyclic peptides and natural peptides, such as mastoperon, which compound has less toxicity and more therapeutic value?
You mean comparison of cyclic peptide? Which can you repeat the question?
Yes, if a comparison is made between synthetic cyclic peptides and natural peptides, such as mastoperon.
Okay, synthetic peptide and natural peptide. Natural peptide, sometimes they have unnatural amino acid. Actually, they have D amino acid instead of L amino acid. We see a lot of cyclic peptide separated from marine organism. They have unnatural amino acid. But those peptides sometimes are very potent and they are much more stable. But experience has shown that they can have very good antibacterial activity and anti-infective activity, but sometimes they are not very selective. They can also kill the normal cell. So we have also used, if you're talking about unnatural natural peptide, cyclic peptide, even we call them natural cyclic peptide. Again, they have many D amino acid, and sometimes they have unusual disulfide bond that cause the cyclization, and sometimes more than one disulfide bond. So in my view, if you rationally design a synthetic peptide using natural amino acid, they are less toxic compared to natural peptide that they have unnatural amino acid like D amino acid. So you can control that. We have also synthesized many cyclic peptides that have unnatural amino acid. Rather than, for example, D amino acid, we use a different type of hydrophobic unnatural amino acid, and we found out that many of them are more toxic compared to natural amino acid. So it's a very hard comparison. So even you have a natural cyclic peptide, sometimes they are much more toxic because the microorganisms generate those peptides to defend themselves and kill other organisms. So, that's why even they may have very good anti-cancer activity, antibacterial activity, and they may be very toxic.
Thank you very much. With this, we conclude our question answer session. We're running out of time, so we cannot take more questions. We have Professor Dr. Iqbal with us. Sir, if you would like to comment Flores for you.
See, my only comment is that it was absolutely fascinating that we have seen every one of the participant attended the entire session. So over 115 people were there from some 30 countries, truly an international event of immense importance. We thoroughly enjoyed an extremely high quality science, and we have appreciated its relevance with the application in drug discovery and development. So my greetings and gratitude to Professor Dr. Pram for not only conducting high quality science, but also to very effectively communicating the science to everyone from a very diverse group of people from different countries. Thank you very much, Professor Pram.
Yeah, thank you so much from you and also all the people who attended this seminar. Thank you so much for coming here.
Thank you very much, Professor, for your time. It was an excellent presentation. And I would also like to thank our audience who had interacted and, was very keen towards your lecture. And with this, we come to end of this lecture. Thank you very much again for all of you for joining. Thank you very much.